Absorption mechanisms can be described as simple diffusion, facilitated diffusion, and phagocytosis/endocytosis. Simple diffusion across the bilipidic membrane barrier is the most important and frequent mechanism of absorption for drugs and toxic agents. Liposoluble non-ionized molecules diffuse down a concentration gradient as a function of their size and partition coefficient. Pores are transmembrane proteins that form a narrow aqueous passage, molecular filter, and that facilitate passive diffusion of water, small ions, or larger hydrophilic solutes across the membrane down a concentration gradient. Channels are transmembrane proteins that form a narrow passage, selectivity filter, across the membrane. A channel can have two conformations, open or closed, due to swiveling of the gate. Channels are usually ion-selective. The flux is driven by the electrochemical gradient across the membrane established by an ATPase, for example sodium-potassium ATPase. Pumps are transmembrane proteins with two gates and one or more binding sites for the substrates. The substrate is bound at one side of the membrane, outside gate open, inside gate closed. This closes the outside gate and the substrate is enclosed within the pump, both gates closed. The inside gate opens and the substrate is released in the intracellular side. Both gates close again to occlude the next molecule. The unidirectional active transport is driven by a source of metabolic energy, most often ATP hydrolysis. Pumps can also work to extrude molecules such as the multi-drug resistant proteins or P-glycoproteins that exude endone xenobiotics out of cells. Carriers are transmembrane proteins with two gates and one or more binding sites for the substrate. The animation shows the simplest type of uniporter carrier with two gates and three conformations. The substrate is bound at one side of the membrane. In this example, outside gate open, inside gate closed. This closes the outside gate and the substrate is enclosed within the carrier, both gates closed. The inside gate opens and the substrate is released in the intracellular side. Both gates close again to occlude the next molecule. The unidirectional passive transport is driven by the electrochemical potential or chemical difference for ionized or non-ionized molecules. Phagocytosis is an energy-dependent mechanism of uptake of large particles, such as senescent erythrocytes, bacteria, parasites and cell debris into large vacuoles, hence phagosomes, by dedicated cells such as phagocytes, leukocytes, e.g. macrophages. Phagocytosis is initiated by cellular contact with an appropriate target, i.e. receptor-mediated, and is energy-dependent, ATP. Receptor-mediated endocytosis is an energy-dependent mechanism of uptake of large molecules, mainly proteins, but also DNA or RNA, occurring in all cells. The first step is initiated by binding of the ligand with the surface receptor. The ligand-receptor complexes accumulate in invaginations of the cell membrane or clathrin-coated pits. Dynamin allows the release of the clathrin-coated vesicles from the plasma membrane and represents the limiting factor for receptor-mediated endocytosis. Examples are LDL-LDL receptor, iron transferrin receptor, anthrax toxin ANTXR1, SARS virus ACE2. Pinocytosis is an energy, ATP, dependent mechanism of non-specific transport for solutes and soluble macromolecules. It is not receptor-mediated. Invagination of the plasma membrane containing solutes results in smaller vesicles than phagosomes or clathrin-coated vesicles.